So, welcome back to it. So, doctor, kinin system we are talking about. What will it do? It converts prekallikrein to kallikrein, and kallikrein is a very important chemotactic factor in the acute inflammation. It will cause the cleavage of a very important substance called as high molecular weight kininogen. It's also called HMWK. When it is being cleaved, bradykinin will form. And who enables the cleavage? Kallikrein. From where is it derived from? Prekallikrein. So, what are the two important functions of kallikrein, doctor? One function is, it is a chemotactic factor, and also it is a one which will basically cause bradykinin formation, is what you have to fundamentally appreciate. Now, what is bradykinin? Bradykinin is once more a peptide containing 9 amino acids, vascular permeability, arterial dilatation. A very important point you need to know, pain, substance P. So, a lot of times this question is asked. Biochemically, what is that important cytokine that mediates the pain response, which is the peptide that mediates pain response? Bradykinin is mainly responsible for mediating pain. It is also arterial dilator and also increases vascular permeability. So, that is the story of prekallikrein becoming kallikrein, which lead to high molecular weight kininogen convert into bradykinin. Bradykinin in turn will lead to the mediation of inflammatory response. Now, few comments on complement system. Invariably questions are asked on complement, doctor. You need to be very sure. Fundamentally, they are the proteins which exist and float, swim inside the plasma proteins. You have enephylotoxins, C3A, C5A. They mediate the degranulation of the basophil, release of the histamine, and the various dirty effects of the histamine they mediate. More than 20 components will be circulating in the plasma, complement system. Then C5A, C3A are enephylotoxins. Then what is C5A by itself can do other than enephylotoxin? Chemotaxis, phagocyte degranulation and stimulation of superoxide radical formation. Then C5b to 9, they increase the cytokine release. Don't remember, nobody will ask about C5b9. They will ask for one reason. It's called terminal complement component. It is called C5b9. It is important for cell lysis. It's also called MAC, membrane attack complex, C5b9. So, suppose if C5b9 is not there, what will happen? Capsulated organisms like pneumococcus, Neisseria, their membrane can't be attacked. So, their infections become florid. Then C3b is responsible for opsonization and potentiation of the antibody response are all the main functions. So, C5a is a favorite MCQ. It is chemotactic. It releases the histamine, increase the leukocyte adhesion molecules. It activates the lipoxygenase system, etc., etc. C3b is an opsonin. C5b9 is called membrane attack complex, is what you need to understand. Then nitric oxide is another important endogenous mediator of inflammation. What is the other name given for it? EDRF, endothelium derived relaxing factor. A lot of times question is asked. What is it chemically? Nitric oxide. Produced by the endothelial cells. It relaxes the smooth muscle. Very important to decide the vascular tone. Now comes a very important question. What is the effect of nitrous oxide on platelet aggregation? Will it inhibit or stimulate? It inhibits, not stimulate platelet aggregation. Lot of times students do a mistake on this point. So, it inhibits. And also, it promotes the thromboresisting property of the vascular endothelium. If good amount of sildenafil is available, nitrous oxide, then thrombus doesn't form. But don't apply this logic while solving questions. Sildenafil is contraindicated if there is a coronary artery disease. Suddenly, if you remember, sildenafil is nitric oxide, and that day, sir, you told that nitric oxide is thromboresistant in nature. So, sildenafil can be given for thromboresistant property to the coronary artery disease, means patient will die first. So, it is contraindicated for a different reason. Why? Because of its vasodilatory nature, it decreases the preload to the heart. Already heart is a failing heart. On the top of it, the guy took sildenafil and wanted to do nocturnal acrobatics, or possibly after taking sildenafil, diurnal acrobatics in the bedroom. What will he do? He will end up in graveyard. So that's the reason, forget about that logic. Basically in pathology, you can't combine too many logics in one place, then it becomes illogical. So it contributes to endothelial thromboresistance is what you need to remember, doctor. Let's quickly talk about the various inflammatory disorders. C2, C3, C5 are the three main complement components. Their deficiency lead to immunodeficiency. Then the defects in neutrophils. Chronic granulomatous disease, you should not forget. What is examiner's favorite question? Inheritance pattern. Very few disorders have X-linked recessive pattern of inheritance. Duchenne's muscular dystrophy. Chronic granulomatous disease also should be remembered because of X-linked nature. You will remember me when you will be solving this question in the real exam. So, in these people of chronic granulomatous, what are the fundamental defects? That NADPH oxidase is there, no? Which is very, very important for that production of the superoxide radicals and hydrogen peroxide formation. That is the one which will basically be deficient in these people. And because of that, if the oxygen burst doesn't occur, phagocytic cells cannot be able to ingest and cannot be able to kill certain microorganisms is what you need to remember. Now comes one of the very important questions of the examiner. You have in microbiology studied catalase positive organisms, catalase negative. Staphylococcus is an example of catalase positive. Streptococcus is catalase negative. So examiner will ask a question. A patient of chronic granulomatous disease, can't he handle catalase positive organisms or catalase negative organisms is the favorite question. Let us see. Staphylococcus aureus like organism, what will it do? It will be generating the hydrogen peroxide during its own metabolism. And the generated hydrogen peroxide is destroyed by its own catalase because it has got a catalase. You agree with me? Then the neutrophils which picked up the organism, they cannot produce hydrogen peroxide because they don't have the NADPH oxidase system available for them. Right? So, the hydrogen peroxide is not available. Why? Whatever the organism produced hydrogen peroxide is already metabolized by the organism's own catalase so that it won't kill the organism. Right? Plus, the neutrophil, the macrophage which has engulfed, it doesn't know how to produce hydrogen peroxide. Agree? So, if the hydrogen peroxide is not available, myeloperoxidase halide system cannot produce a halide superradical. HOCl it can't produce. If that is not produced, the organism is not killed. So that is the reason these people can't be able to handle catalase positive organism like staphylococcus. If they infect, they can't be killed. They can be engulfed by the macrophage, but they can't be killed. Then how about catalase negative organisms? Catalase negative organisms during its metabolism will produce H2O2. Right? And what will happen then? They don't have catalase to break it down. Right? Then even though the macrophage doesn't have hydrogen peroxide, the hydrogen peroxide produced by the bacteria, which is not detoxified by the catalyst, because organism Streptococcus doesn't have catalyst. That hydrogen peroxide will be used by the myeloperoxidase system in the macrophage, and as usually the superoxide halide radical is produced, and that will kill the organism. That way, who is killing the organism? Suicide. Its own hydrogen peroxide, which is not metabolized because it can't produce catalase, will help the macrophages myeloperoxidase to use the bacterial hydrogen peroxide and produce a HOCl and kill the organism. See? So there is a reason Streptococci can be handled, but not the Staphylococci is what need to be remembered. That's the reason, doctor. Chronic granulomatous disease. When tomorrow examiner asks a question, what will you answer? Catalase positive organisms like Staphylococci can't be handled. Infections will occur, but catalase negative organisms like Streptococci will be normally killed. Right? So wherever logic is involved, don't apply mnemonics. You will get a irreversible pneumonia. So you need to apply logic. Right? Where brute memory is required, you can use it. If it is CA or CB, someday you need to remember, like a CID, what is, which is anaphylatoxin, which is chemoattractant. There no logic, don't apply logic there. There you use some sort of masala mnemonics. But where logic is required, Staphylococci, why it is not killed, Streptococci, why it is killed in chronic granulomatous? Yes, you can still remember it through logic. Then comes the myeloperoxidase deficiency. In these people, what will happen? Organism is taken up, superoxide H2O2 is formed, but that H2O2 cannot be combined with the halide system to produce HOCl in the people with myeloperoxidase deficiency. But they will not have infections as bad as chronic granulomatous disease, is what you need to remember. And but one important point, favorite MCQ is susceptibility to candida infection. Very, very high in the people who are having myeloperoxidase deficiency is what need to be remembered. Now, doctor, Chediac Higashi. Chediac Higashi is autosomal recessive in nature. There are some phenotypic features of the Chediac Higashi which you should not forget. Neutropenia, albinism, cranial and peripheral neuropathy. These are all the important associated features. They are not asked as an MCQ. Nobody bothers, because we are not interested about what is the syndrome. We are interested about what are the problem, immunological problem. Okay, what is the immunological problem in Chediac Higashi? In them, there will be repeated infections, very characteristic feature. If you take their peripheral smear, a huge, abnormal WBCs, multilobated, is a very, very important abnormality you find. Then what is the problem in the Chediac Higashi people? It's not with superoxide radical formation or with the myeloperoxidase halide system. In them, there is an abnormal microtubule formation. If the microtubule is not there, the leukocyte become lazy. Even though they are calling, there is a theft, please come, there is a theft, please come. Police officer has fully drunk on that night duty. He doesn't want to wake up at all. Lazy leukocyte. Because of the improper microtubule formation, it can't be able to move. So that is a problem in Chediac Higashi, is what need to be remembered. So what are the important morphological abnormalities in Chediac Higashi? Typically, there is a problem in fusion of the lysosome with the organism which is being phagocytized inside the macrophage. There will be large cytoplasmic granules in the granulocytes, and similarly there will be abnormal melanosomes in the melanocytes. Everywhere there is a laziness. Melanosomes don't want to form, leukocyte doesn't want to move, all because of the microtubule problem. And because of this melanosomal formation abnormality, what is the important phenotypic expression? Albinism, neutropenia, cranial and peripheral neuropathy are the important associated features is what need to be remembered. Last but not the least in acute inflammation, leukocyte adhesion deficiencies, LADs, is another important. We already, where is LAD1, LAD2 factor? You still remember? Just before we studied, no? The integrins, selectins, cadherins, etc., etc. There that LAD1, right? So LAD1 is called integrin. Some people can have a deficiency. LAD type 2 deficiency may there is a gene which has an abnormality with that CLI Lewis X on the neutrophils, which will enable it to bind with the vascular endothelium, leukocyte and vascular endothelium. That is deficient in LAD type 2 deficiency. So that is all the story, doctor, of this most cruel topic called as inflammation, which is hypnagogic hallucination in the mind of a imagining medical student.